Mitosis. You know, mitosis is an equational division. Here, they are responsible for increasing the size of an organism. The chromosome number in the daughter cells remains the same. Division, mitosis or equational division. Mitosis in plant cell is of anestral type, that is, it lacks centriole and estral rays, and mitosis in animal cell and few lower fungi is of estral type, that is, it is characterized by the presence of centriole and estral rays. Mitosis produces genetically identical cells. These are similar to the mother cell. Cell cycle. Complete life cycle of a cell is called as cell cycle, and cell cycle is divided into two stages, that is interphase, and the second is the division phase. Now let us take up the interphase. You know, it is one of the most important phase of the cell cycle. Middle stage of the cell cycle, there occur one interphase between two division phase. In interphase, cell grows in size and prepare itself for the next division. It is the most active phase of the cell cycle. Metabolism of the cell increases, and it is divided into G1 phase or gap phase 1. It is one of the longest phase of the cell cycle, and if you take 24 hours, so it takes about 12 hours time. During this phase, cell rapidly synthesizes different types of RNA and protein. Due to availability of protein, synthesis of new protoplasm takes place in the cell. S phase, synthetic phase, it is taking 6 to 10 hours, and here you will see that the duplication of the DNA takes place. It involves replication of nuclear DNA and synthesis of histone protein. In structure of chromatin threads, following changes take place during the S phase, that is the duplication of chromatin, replication of the DNA, and a special protein, cyclin, which triggers the DNA replication, is also synthesized. G2 phase or gap phase 2, it varies from 3 to 12 hours. Number of the cell organelles increases in cell. Cell prepares itself for the cell division during this phase. Special material required for cell division are synthesized during this phase. For example, tubulin protein required for the formation of a spindle fiber. And in addition to that, cell division also involves enormous expenditure of energy. So, energy is stored in the form of ATP. After G2 phase, cell enters the division or M phase, and the duration of M phase is 1 hour. Now you can see the entire cell cycle. You have G1, S, G2 and M. That is the gap phase 1, followed by replication of the DNA, duplication of the DNA, and the cell preparing itself for the mitosis in the gap phase 2. Division phase. Division of nucleus occur by sequential changes in nucleus and cytoplasm. You know, the first phase is the prophase, and here you will see the cytoplasm becomes viscous and refractive. Chromatin threads condense, that is, they become thick and conspicuous due to loss of water, and they become distinct chromosomes. In animal cell, as I told you, that it is of estral type, the centriole starts moving towards the opposite pole. Estral rate formed due to dilation of protein around the centrioles. And of course, by the time the cell enters the late prophase, the nuclear membrane disappears and the nucleolus disorganizes. You can see the nuclear membrane is disappearing and nucleolus disorganizes, and the condensation of the chromosome has taken place. Metaphase. Chromosomes, they come to lie at the equatorial plate to form a metaphase plate or metakinesis. chromosomal fibers, that is pole to centromere, and supporting fibers, that is pole to pole, or you can say full length fibers arranged in the cell. Each chromosome is splits lengthwise up to the centromere, and the two chromatids of a chromosome repulse each other and move towards the opposite pole. This is the metaphase. You can see the chromosome lying at the equatorial plate. The centromere has divided and the chromatids will start moving in the next phase. Anaphase. In early anaphase, interzonal fibers appear at the equator of cell. In late anaphase, chromosomal fibers contract and interzonal fibers expand, so chromatids move to opposite poles by pull and push mechanism. And you see in the diagram, the movement of chromatids to the respective poles is the beginning of the anaphase, and you can see in the third diagram that they have reached to two poles almost. Telophase or reverse prophase. Metabolism of the cell starts increasing. Nuclear membrane reappears around the chromatids and the nucleolus reorganizes. The chromosomes decondense and they form the chromatid. You have seen in prophase condensation has taken place and now they decondense to form the chromatid. Now you can see the telophase. The chromosomes have reached to the two poles, and of course in one diagram you can see in the animal cell the constriction has also started. Cytokinesis is the division of the cytoplasm. Cytokinesis in animals, the cytokinesis is taking place by constriction and furrow formation. Microtubule and microfilament arrange on equator to form a mid-body. Contraction occurs in the mid-body and the plasma membrane starts constricting and a furrow starts forming from outside to the inside of the cell. And you can see in the first diagram that almost the furrow is complete and they are going to form the two daughter cells with identical DNA. Then the rest two diagrams are showing the cytokinesis in plant cell. In plant, cytokinesis is by cell plate formation due to presence of a rigid cell wall. Numerous Golgi vesicles arranged on equator to form phragmoplast, endoplasmic reticulum and fragment of spindle fiber also collect on equator to form a cell plate. Golgi vesicles secrete calcium and magnesium pectate, which helps in the formation of middle lamella, and in plant, cytokinesis occur in a centrifugal order. Now you can see that from the middle, the cell plate starts moving towards the two ends, and that is how the cell plate formation is taking place in the plant cell, while in animal cell you have seen it was by constriction. Modification of mitosis. Dinomitosis. It is somewhat different in the sense because it occurs only in dinoflagellates. Cell division of dinoflagellate. Dinoflagellates are mesokaryotes. You are aware of the two terms, prokaryotes and eukaryotes, and it occupies a position between the two, that is the mesokaryotes. Due to the absence of histone proteins, chromosomes fail to condense properly, so chromosomes are not distinctly visible during division. Nuclear membrane present throughout the division. As you have seen that the nuclear membrane at the late prophase disappears, but here you will find that the nuclear membrane remains throughout the division. Endomitosis. Division or duplication of chromosome without the division of the nucleus. Endomitosis leads to polyploidy, and this you can find very common in tapetal cells, and that is why you will see the tapetal cells show the polyploidy.